Thank you for taking the time to view our presentation. All of the team members here at Chromacell welcome the opportunity to present our potential breakthrough therapy for erythromelalgia and to share new and exciting work with COVID-19. My name is Kenneth Kashkin, and I am the COO of Chromacell. I have over 30 years experience in the pharmaceutical and biotech industries, with executive experience from companies such as Abbott Labs, Baxter, and several biotechs with a successful IPO. I have been with Chromacell for about 5 years with the prime responsibility of growing our therapeutics division. I have the honor of presenting you with an overview of the exciting products we have available for partnering or investment. I would like to note that if you have any questions or would like to speak further about the presentation, please reach out to myself or Christian Copley, our CEO, at the email on the current slide. Chromacell was founded in 2002 with a focus on therapeutics and flavors. The main objective of the company was to leverage our proprietary technology, ChromaVert, to create cell lines which express difficult targets. The company has a strong base business in flavor research and has continued to pursue therapeutic targets using the ChromaVert technology. As the therapeutics division of Chromacell took shape, we leveraged this groundbreaking technology to help drug hard-to-express targets and enable us to take a leading role in the development of drugs targeting sodium voltage-gated channels. Currently, we have a lead molecule that is ready for phase 2 proof of concept that targets NaV1.7, and we are working on using our technology and prior research to spur new developments in the COVID-19 space. The team at Chromacell has a wealth of knowledge and extensive experience in management, discovery, and development, and we would welcome the opportunity to interact personally at a future meeting. Chromacell's lead molecule, CCA464, has shown a preclinical and clinical profile that makes it an ideal compound to enter a proof-of-concept trial in the rare disease of erythromelalgia. 8464 has shown robust preclinical efficacy in a large number of animal models, ranging from chemotherapy-induced peripheral neuropathy and diabetic neuropathy models to models of inflammation as well as postoperative pain. It is peripherally restricted, meaning that we do not expect to see a problem with CNS-related adverse effects or any dependency concerns. In fact, we have never seen these effects in animals or in our phase 1 clinical studies. 8464 has also gone through significant CMC development and has already been produced in greater than 30 kilogram batches, with sufficient drug substance and product available to support any initial clinical aims. In addition, 8464 has completed toxicology studies with up to 90 days of treatment in multiple species, with NOELs giving a large safety margin over our anticipated clinical doses. In the clinic, we have seen 8464 exhibit a tolerability profile that clearly supports the proposed erythromelalgia proof-of-concept trial design. NaV subtype 1.7 itself is considered a validated target for pain therapy. This is because mutations in the SC9A gene are linked to so-called gain-of-function disorders that increase pain, such as erythromelalgia and paroxysmal extreme pain disorder, whereas loss-of-function mutations result in congenital insensitivity to pain. This lack of pain-associated mutation-related inhibition of function has validated NaV1.7 as a target for analgesia. CCA464 is the most potent and selective NaV1.7 inhibitor in clinical development. Its in vitro potency and selectivity echoes that of Pfizer's prior NaV1.7, but 8464 may be as much as 4 times as potent and have more effective binding kinetics, which should allow for stronger target engagement. In a chronic dosing rat model of diabetic peripheral neuropathy induced by STZ, 8464 shows the ability to reduce both thermal hyperalgesia and tactile allodynia with almost complete elimination of pain, while simultaneously displaying no adverse effects, including CNS-related. Most recently, Chromacell has also investigated 8464's ability to prevent chemotherapy-induced peripheral neuropathy in a rat model. We used oxaliplatin as the chemotherapy agent and treated the rats with the chemotherapy and 8464 in tandem. 8464 showed a dose-dependent ability to prevent the emergence of the neuropathic pain. Additionally, when looking at the literature, another strong NaV1.7 tool compound, Protex2, was shown to also prevent the development of peripheral neuropathy in a taxane model of CIPN. We believe our ability to replicate this published data in a different model of CIPN shows strong target engagement and that NaV1.7 has the potential to prevent CIPN in humans. In the clinic, 8464 has completed 4 phase 1 studies, including 2 relative bioavailability studies to test new formulations, an SAD-MAD study that treated subjects up to 14 days, and a warfarin DDI study. We saw no effects on vital signs, olfaction, LDL levels, or lab values, nor the presence of cardiac or CNS events. The only clinically relevant dose-limiting study safety finding was a dose-dependent drug-induced rash, which was seen in our SAD-MAD study and our DDI study. As with other drugs exhibiting this side effect, desensitizing dose escalation for chronic administration will be able to reduce the incidence to what we expect will be as little as 2 to 3%. We believe that 8464 is uniquely positioned to address the rare neurovascular disease known as erythromelalgia. This debilitating disease usually presents in the feet and or hands and may emerge as early as childhood. It consists of intense pain and erythema flares, which are triggered by warmth, physical activity, or stress. The threshold for a flare could be extremely low in some patients, with simple acts such as putting on socks or shoes being enough to initiate an extremely painful flare. Patients with more severe cases have an abysmal quality of life, which often leads to depression, anxiety, and suicidal tendencies. There are currently no approved therapies in erythromelalgia, and no delivered treatment has been shown to be effective in this population. Erythromelalgia occurs in up to 50,000 patients in the US, and current estimates of NaV1.7 genetically mutated patients is approximately 5,000. Looking at the market opportunity, we have used non-24-hour sleep-wake disorder to represent a similar market size. For this condition, Hetlioz has sold for about $200,000 per year per patient. Using our best estimates of patient epidemiology and penetration, we believe 8464 has the potential for sales in the $750 million to $1.25 billion per year range. Prior NaV1.7 targeting drugs, including the Pfizer compound and the Xenon compound, have been tested in small pilot studies of erythromelalgia with impressive results. These drugs were later discontinued due to safety signals and lack of tolerability. Here we present the data from Pfizer's pilot study in 5 subjects with NaV1.7 mutated erythromelalgia. As you can see, they had robust changes in the NRS at 4 to 9 hours post-dosing when compared to placebo, with an impressive statistical signal from such a small n. In addition, data is available for the Xenon compound, which also showed robust efficacy in a very small patient number. We believe the fact that other NaV1.7 targeting compounds have shown impressive efficacy data in humans in this extremely difficult to treat population gives us a high chance of success for a positive proof of concept trial. Currently, we are planning a randomized double-blind crossover study in 10 NaV1.7 mutated erythromelalgia patients to show proof of concept. This study follows the advice of Dr. Stephen Waxman, professor of neuroscience at Yale, the leading NaV1.7 expert in the world, who conducted many of the previous erythromelalgia trials. We believe this study has a high chance of success and can be performed in a rapid manner in the United States. We believe that 8464 will receive a breakthrough designation, and with that in mind, we can reach the market as soon as 2023. We expect that the medical need and its rare occurrence will support a streamlined approval pathway with post-approval commitments. In addition to CCA464, we are also working on using our ChromaVert technology to allow for key research and development into COVID-19. We believe that our technology will allow us to create stable cell lines that express cytotoxic viral proteins. These cell lines could be vital in drug and vaccine development, and we have a strong desire to join the research community to help end the threat to our society that is COVID-19. With that in mind, Chromacell has evidence from its expertise and research in the ENaC space that may benefit COVID-19 patients, and we are looking to drive this research forward into human trials as soon as possible. It is known that epithelial sodium channels are expressed in lung alveoli and regulate fluid clearance. Through Chromacell's prior screening, we have identified some ENaC potentiators, both novel and commercially available drugs, which may represent an advance in the treatment of COVID-19, and we are excited to begin working towards an IND and clinical trials. Chromacell is a well-established and strong biotech company which has the capability to discover and develop new therapeutics that can change the landscape for patients facing erythromelalgia and other diseases. We believe that our capabilities, expertise, and products represent a strong partnering and our investment opportunity, and we would welcome the opportunity to talk more in depth about any potential relationships. Please reach out to Christian Copley at your earliest convenience to further any interest or discussion. Thank you so much for your attention and consideration.